Mm, A very good afternoon to Professor Hemant Kumar sir. So I hope that my voice is audible to you and to all the participants. Am I audible?
Yes, ma'am.
Yes, ma'am.
Professor Hemant sir, may I start? Hemant sir, am I audible to you?
Yes, you're audible.
Okay, thank you, sir. So with your due permission, I would like that I would start the session. So good afternoon to everyone present here. I, Deepti Goyal, Assistant Professor in Department of Physics, would like to welcome each one of you on this beautiful auspicious day of the second day of the International Conference on Integrative Chemical Science for Health and Environment. It's my pleasure to introduce Professor Hemant Kumar Rajor, sir. He is a professor in Department of Chemistry, Ramjas College. He is a Raman Fellow recipient and he has done his postdoctoral research from San Diego State University, San Diego, California, USA in 2017. He is also a recipient of Young Investigator Award of American Chemical Society to participate in Gordon Research Conference on Green Chemistry in 2004 in USA. His research areas and interests are in coordination chemistry, catalysis chemistry, nanocomposite, wastewater treatment. He has many national and international collaborations and along with long list of peer-reviewed research publications and research projects to his credit. He has given his research guidance to many graduate and undergraduate students, and he has dedicated his 23 years of teaching fraternity. He had shared many administrative responsibility and even served as the vice principal of Ramjas College for 2 years. And he has immensely contributed to the corporate life of the college. His dedication and expertise promise to significantly impact the academic and research landscape, contributing to a better and healthier world for all of us. I welcome you, sir, and I again welcome you as the chairperson of this session. I welcome you, sir.
Yeah, thank you, Dipti.
Yes, sir. Now I'm going to show the guidelines for the oral presentation for all the participants.
Yes, Dipti, you will give some guidelines or what? How many time they will take?
Yes, yes, sir. I'm just showing the guideline. I'm sharing
Yeah.
the screen and showing the guidelines to them. The guidelines for the oral presentation is that the participant will be given 8 minutes and extra 2 minutes for the presentation. Means a total of 10 minutes will be given them for the presentation. The participants will unmute only once his or her turns comes. Participants are requested to sit at a silent place and prefer earphones for not any disturbances. Participants have to attend the complete session. Participants have to turn on their camera during their presentation, and evaluation will be done by the selected panel of judges. So I hope that the guidelines is clear to all the participants. Now I am sharing the screen of the list of participants. I hope that from the screen everybody has joined, all the participants have joined, and we shall carry out the presentations in the order of their number itself. So let us take the first call of the participant. The first call is for the Surbi Goyal. She is a PhD research scholar, and the topic of her presentation is analysis of good and bad fingerprint for identification of NIR based optical frameworks using Monte Carlo method. Surbi Goyal. Surbi Goyal, are you here? Surbi Goel, are you able to hear me? OV91. Surbi Goel is not
Good evening.
okay. Okay.
Good evening, ma'am.
Yes, Surbi, now you can take over and you can share your screen and you can start your presentation. Hemant,
Okay, thank you, ma'am.
sir, anything from your side?
Aditi, you can go ahead.
Yes, sir.
Surbi, you can start.
Yes, yes. Surbi, you can start now.
Ma'am, is my screen visible?
No, it is not visible. Surbi, your presentation is not visible.
Sir, I am trying. Sir, is my screen visible?
Yes.
Okay, thank you, sir. Good evening to all. I am Surbi Goel from PhD Research Scholar from Department of Chemistry, BMU Rohtak. Today, I will present my work on Coral software. The title of my presentation is Analysis of Good and Bad Fingerprint for Identification of NIR-based Optical Frameworks using Monte Carlo Method. First, introduction. QSR is a computational modeling method for developing mathematical correlations between the chemical features and the behavioral manifestations of structurally similar compounds. Development of QSR model plays a significant role in the design of purpose-specific fine chemicals, including pharmaceuticals. QSAR analysis is now used for the prediction and design of chemicals of health benefits, industrial or laboratory process, or household applications. QSAR analysis employs chemical information from a small number of chemicals, deriving a mathematical correlation while allows a prediction of same response for a large number of chemicals. The basic formalism of QSAR technique can be mathematically represented as follows. Biological activity is the function of chemical attributes. The QSAR study comprises of the QSAR study comprises of The QSR study comprises of three simple steps. First is data preparation, that is arrangement and conversion of the data in a suitable form. Second is data processing, that is includes data pre-treatment and development of suitable model. Third step is data interpretation, which includes prediction of the response of existing untested or new chemicals and designing of completely new chemicals possessing the desired activity. This is the flowchart for the QSR modeling. These are the molecules which we can take for QSR modeling.
Surbi, your slide is not moving. You are still on introduction phase.
Okay, sir.
technical problem?
Ma'am, I don't know what is the problem. Actually, I tried on laptop, but its speaker is not working.
Your voice is coming, but the slide is not changing. You are just
Ma'am, actually, I am using two devices. One is phone and another is laptop.
uh,
But
from the one you are
um,
displaying your PPT. Please double click on that 2 3 4 ना, तो आपका आ जाएगा.
ma'am, 1 minute. Ma'am. Ma'am, is my screen visible?
Yes, yes, beta. Yes, now it is changing.
Okay, sorry, ma'am, for the inconvenience. Can I start, ma'am?
Yes.
Yes, sir. So this is a flowchart for QSAR modeling. First, we take a number of compounds like ethylbenzene, nitrobenzene, chlorobenzene, etc. Then next step, we calculate the descriptors like molecular weight, number of carbons, number of nitrogens, number of oxygens, etc. Then in next step, data pre-processing is done, in which we do normalization, standardization, feature selection, identification of outlier. And then in next step, a table is prepared in which in first column we take the compounds, and then in next columns we take the descriptors, and in last column we take their activity value. Then we apply the statistical tools of QSAR. That can be MLR, that is multiple linear regression, PLS partial least squares, ANN artificial neural network, SVM sport vector machine. Then after the preparation of model, we does their validation via various statistical metrics like R square, Q square, R square predicted, or RMSE, etc. Then we plot a graph between the predicted activity and the observed activity. And the graph we obtain should be a straight line. It shows a linear relationship. Now, what are the I work on NIR based chromophores. So, what are the applications of NIR based chromophores? These chromophores are widely used in bioimaging, photodynamic treatment, optoelectronics, sensing, and anti-counterfeiting applications. Their advancement has significantly increased the potential for non-invasive imaging, targeted therapy, and optical communication systems. Medicine, biology, material science, and telecommunications have all benefited from this progress. Now, we can see from the diagram the applications of NIR-based optical materials. They can be used in imaging and diagnostics. They can be used in photodynamic therapy. They can be used in optoelectronic and sensor, and in anti-counterfeiting. Now, steps to design NIR chromophores. I designed an NIR chromophores by the following steps. In first step, I prepared a dataset of 292 chemicals with their absorption maxima in ethanol. Then I developed QSAR model whose R-square validation was 0.8275. Then I does the mechanistic interpretation of the model and extracted the parameters of increase or decrease of the endpoint. By these attributes, I designed 4 new chromophores with absorption maxima 791 to 822 nanometer that lies in the NIR region. The first step is QSAR modeling. I prepared the model of QSAR by using Coral software. I divided the dataset into 4 sets, that is training set, invisible training, calibration set, and the validation set. And the model equation is shown in the table. The model was very robust, predictive, and reproducible. This can be seen from the statistical characteristics values like R-square value was 0.9459, IIC was 0.7410, Q-square value is 0.9428 and mean absolute error was just 13.6. Now, this is the graphical representation between calculated lambda max and the experimental lambda max. Now, from the graphs, it can be seen that it shows a linear relationship between the calculated value and the experimental value. Next, the graph is between the residual lambda max and the experimental lambda max. Residual lambda max is the difference between calculated activity and experimental activity. Now, next step is mechanistic interpretation. In the mechanistic interpretation, we extracted the structural attributes. That is the attributes which increase the lambda max and which decrease the lambda max. The attributes that can increase the lambda max are one, that is presence of one ring, presence of two rings, presence of nitrogen with double bond, presence of double bond, association of nitrogen with carbon that are sp3, presence of double bonded carbon, and association of double bonded bond with oxygen and branching. Now, I design the chromophores like this. I take the lead compound with value 726 nanometer and then inserted the fragments that increase the lambda max. For example, I inserted in this compound the double bond, 3 aromatic rings and nitrogen. And we can see that the lambda max value was was increased up to 813 nanometer. That lies in the NIR region. In the next molecule, I inserted the double bond, nitrogen, aromatic rings, and the methyl group. That causes the red shift up to 822 nanometer. So I designed these NIR chromophores, which can be used in which have various applications. Now the conclusion. Coral software is utilized to model a dataset of 292 organic chromophores to assess their absorption maxima. Monte Carlo optimization along with target function 2, that is index of fidelity of correlation, was employed to develop models that exhibit high robustness, predictability and reproducibility with R-square validation of 0.8275. Then the mechanistic interpretation was employed to assess promoters of increase and decrease of absorption maxima. Utilizing positive attributes, 4 new NIR-based chromophores were designed, which can be used in the development of NIR-based imaging agents. It is possible to use the newly designed chromophores for the development of NIR-based optical materials using organic chromophores. This study opens new horizons in computer-aided optical material development and proves to be a substantial advancement in the field of QSR development. These are my references I used in my work. Thank you all.
So the session is open for the question answer.
Yes, participants can ask question. Surabhi, you can exit from this presentation.
Okay, sir.
So that participants, you can ask any question if you have. No one is interested in to ask any question. Okay, so then I will ask. Surabhi, this is your which year of research? Surabhi Goel, can you hear me? I don't know whether. Surabhi, can you hear me?
Surbi, kindly unmute yourself.
Surabhi Goyal, can you unmute your mic? Yes. Surabhi, tell me, you have used this Monte Carlo method, right? So why you are not using other methods also? Because there are other methods are also available. Why you have chosen only Monte Carlo method? Surabhi, do you understand what I'm asking? So because time already lapsed, so can we move to the another participant? Deepti, can we move to another participant?
So I move to the next participant. The next participant is Manisha. Her topic is
Yes,
DFT,
ma'am.
DFT study of molecular electrostatic potential, reactivity, and optical properties of derivatives of selenophane fused pentacene isomers. So Manisha, you may take over. Yes, your screen is visible, Manisha.
Ma'am, sorry to interrupt. Am I audible?
Surabhi, your time is already lapsed. So sorry, we have to move to another candidate, Manisha.
Good evening, all of you. I am Manisha, a research scholar of Baba Masnath University in the guidance of Dr. Vijay Dangi and Dr. Varamdat Arya. Today, I am here to present my research work on DFT study of molecular electrostatic potential, reactivity, and optical properties of derivative of selenophane fused pentacene isomer. Now, introduction. Due to lightweight and more flexible nature and low cost of organic semiconductor materials than or inorganic semiconductors, materials make them alternative use of inorganic semiconductor. The performance of organic semiconductors depend on the charge carrier mobility in the organic compound used. Therefore, the mobility of the charge carrier in pentacene molecule is more as compared to other organic materials. Therefore, the pentacene derivative are widely used in studied organic materials for use in organic semiconductors. Pentacene compound fused with selenophane rings are widely used to increase the performance of electronic devices such as organic semiconductors. These compounds are made up of carbon, hydrogen, and selenium atoms. Pentacene compound fused with selenophane rings improve charge carrier mobility, which leads to small energy gap and increase the conjugation length. Therefore, attaching the electron withdrawing groups such as chloro and cyano group to the pi conjugated rings, which improve the charge carrier mobility. Now these are the reference review used ICHE-2023 compare the neutral anionic and cationic form of oligo and polyselenophene with polythiophenes. They found that oligo and polyselenophene have lower band gap and more charge carrier mobility than the polythiophenes. ICHE-2023 also studied the electron transport properties of the pi organic conjugated molecules, which attaching with the electron withdrawing groups and found that improved electron transport properties. Now the objective of work. Discuss the electrophilic and nucleophilic site by using the molecular electrostatic potential surfaces. Observe the impact of change, the position of selenium atom and substitution of chloro and cyano group on energy gap and electrophilic character and absorption maxima. Research process. The Gaussian software package and DFT methodology was used for all the calculation. Firstly, ground state optimization of all these studied selenophenol rings fused pentasine isomer is done by using the DFT method, and then calculate the further different properties like absorption maxima, molecular electrostatic potential surface, HOMO-LUMO energy gap, molecular electrostatic potential map. It is helpful to determine the nucleophilic and electrophilic center of a compound. Here, blue color shows the low electron density and a nucleophilic attacking site, and red color shows the high electron density and electrophilic attacking site. Therefore, green area shows neutral part and zero potential. These are the molecular electrostatic potential map this studied compound. Here, impact of position change and substitution of electrophilic character. Increase electrophilic character increases with increase in electronegativity and electrophilic index, while decrease in chemical potential with change in the position of the selenium atom and attached chlorine and cyano group. Now, impact of position change and substitution on the energy gap, hardness, and softness. Energy gap between HOMO and LUMO decreases with the change in the position of selenium atom and attached chlorine and cyano group, in which compound become more softer and less harder. This tends to stability decrease while reactivity and charge carrier mobility increase. Impact of change, position change and substitution on absorption maxima. If change the position of selenium atom, blue shift observed, and if attach the chloro and cyano group, red shift observed. And the range of this study compound lies between 451 to 635 nanometer, which lies in the visible region. Now the conclusion. According to the molecular electrostatic potential surface, dark red color and high electron density shown on nitrogen atom of cyano group, and electrophilic character is more predominant, and red shift is observed with change in the position of selenium atom and attaching of cyano group. It is also observed that softness increases and hardness decreases, which increases the charge transfer process and reactivity with change in the position of selenium atom and attached chlorine and cyano group. These are the reference used. Thank you.
Yeah, any participants? Do you have any questions for Manisha?
May I ask one?
Participants, you can ask question from Manisha.
Manisha, can you elaborate what do you mean by HOMO and LUMO?
Hello.
Hello, Manisha. Can you elaborate upon what do you mean by HOMO and LUMO bands?
Hello. Energy gap between the HOMO and LUMO orbitals, that is FMO orbitals, energy gap between them.
No. What is the full form of HOMO and LUMO?
Higher occupied molecular orbital and lower unoccupied molecular orbital.
Okay, so you have used DFT, right?
Yes, sir.
So, can DFT is useful tool to analyze data for a larger molecules?
Yes, sir.
Sure.
हाँ जी सर.
So which software you are using?
Sir, Gaussian software.
Okay. Okay, then fine.
Thank you, sir.
Any participants, if you wish to ask, then you can ask. Otherwise, we will move to the another candidate. Dipti,
Yes, yes, sir. So thank you, Manisha.
yes, we can move to another.
Next is Yudhishthir Vaishnav.
Thank you, Manisha.
So Yudhishthir Vaishnav's topic is to synthesize of amino acid derivatives of 1,3,4-oxidazole and their use in drugs. Yudhishthir Vaishnav, you can start your presentation.
Yudhishthir Vaishnav. Dipti, next one is Yudhishthir Vaishnav, right?
Yes, sir. Yudhishthir, please.
But voice is very slow. I don't know why.
So my voice is low, or the participant's voice is low.
Now your voice is okay, Dipti.
Okay. So Yudhishthir, please take over. Yudhishthir, you may start your presentation. Yudhishthir, your voice is not audible. You have not unmuted yourself, Yudhishthir. Yudhishthir, unmute yourself and then speak. Sir, is my voice audible to you?
Yes, Dipti. Yudhishthir, your voice is not audible. Is there any problem, Yudhishthir?
Sir, is it audible? Hello.
Yes.
Hello.
Yes,
Can you
Yudhishthir,
hear me,
you are
sir?
audible.
Okay, sir.
Yes, of course. We are.
Sorry for inconvenience, sir.
Okay, start, please.
Hello. Okay, sir. Sorry for inconvenience. I'm from Jodhpur. My name is Yudhishthir Vaishnav, research scholar, and my topic is to synthesize of amino acid derivatives of 1,3,4-oxadiazole and their use in drugs. So the abstract, which is the work done on this topic, actually I use a heterocyclic compound and as like amino acid and prepare some derivatives of amino acids using some acids and some other compounds. And the study is done on the antibacterial and antimicrobial property on the compound which is synthesized by me in my project, my work. And some other medicines are also used for reference to study of antimicrobial and antibacterial properties like ciprofloxacin, fluconazoles are also used. And this is introduction about that what is oxadiazole, and it is a heterocyclic compound, 5 member zinc, and the substituted oxadiazole is prepared. 1,3,4-oxadiazole, the derivatives are prepared. My research work, and using amino acid, substituted oxadiazole is produced, and after that, its activity is studied by me in on antibacterial and antimicrobial phenomena. The material which is used both of reagent and solvent were obtained from commercial sources, and the capillary technique was used to get all of the melting point, and the IR spectra using Perkin Elmer FTIR spectrophotometer, and chloroform is used as a solvent collection for HNMR spectroscopy, and this is synthesis and characterization. actually three types of amino acid, aromatic amino acids like tyrosine, phenylalanine, tryptophans were used to synthesize amino acid derivatives of 1,3,4-oxadiazoles, and after acylation of it, and the following procedures after using hydrazides in presence of iodine and potassium carbonate used for formation of 1,3,4-oxadiazole derivatives. And this is the reactions which are followed by the formation of the product. And this is scheme first, and this is scheme second by phenylalanine and succeed used by me and laboratory, and data IR spectras and NMR spectras are calculated. And the antimicrobial evaluation is done on gram positive as well as a gram negative bacteria. Gram positive just like Staphylococcus aureus and Bacillus subtilis, and gram negative like Escherichia coli and Pseudomonas aeruginosa, and fungal species just like Aspergillus niger and Candida albicans were taken for antifungal examination. The sample is used 100 microgram per ml, and a reference sample used for 20 microgram per ml. These are the drugs which are already present in market for comparative studies. Now these are the data, and whatever the data obtained by the study, they are static in form. And result and conclusion that after the all of the work I have calculated, and we decided is some the product the derivatives product produced by in our my project, some of that like A1, A3, B1, B3, B2, B3, which are on in these slides, previous slide, they are 75 to 95% more effective against Staphylococcus aureus and Bacillus stabilis, and some are 60 to 65 to 70% effective, and on Escherichia coli they are 75 to 95%. And these are the data and conclusion result after my work. So it has proved that some of the derivatives of oxadiazole are very useful for antibacterial and anti-fungal activities. These are the reference. Thanks to all from Yudhishthir West.
Thank you,
Hello.
Yudhishthir.
Yes, participants, you can ask. Any query if you have? Participants, yeah, it is supposed that you should ask some question from the presenter. So anyway, Yudhishthir, in your research, you have mentioned that you have analyzed your compound by various methods like proton NMR, like IR, and CHN studies also, right? What is CHN?
Yes, sir, yes.
How you analyze CHN?
Sir, actually the sample was sent in a defense lab, and there are some my friend, they give me data after the testing of my metal, which is prepared by me in a lab.
Okay, with one thing in proton NMR, you use
Yes,
TMS, right? What is the full
Yes.
form of TMS?
This is a tetramethylsaline
So
reference. This is used as a reference.
can we use some other chemicals also in place of TMS?
No, sir. Actually, in TMS, tetramethylsaline, all of the 12 hydrogens are chemically and magnetically equivalent. So the peak is obtained at a 0. But we can use it as a better reference.
No, better, but can we use some other reference chemicals also?
Sir,
in
if
place
it
of
is
TMS?
not interfering, if is it not interfering in our sample, then can we use the? But I don't use the. I use a TMS, sir.
Okay.
If it हाँ sir.
So you use only proton NMR. You have not used G-COSY
हाँ.
and something like that?
No sir, until I have not used sir.
Okay, so from where you have analyzed your sample by proton NMR?
Sir, it is a defense lab and defense lab of Jodhpur.
Okay. Okay, fine. Anybody, if you wish to ask, so you may ask. Otherwise, we may shift to another one.
Thank you, sir. Thank you
Thank
very
you,
much, sir, for
Yudhishthir. Thank you.
give me precious time. Thank you, sir.
Okay, Deepthi, we can
Thank
move
you,
to another
Yudhishthir.
one.
Thank you. You may
Thank you very
stop
much,
your
ma'am. Thank you. Thank
screen
you
sharing,
for
Yudhishthir. And the next
okay.
participant is Kamal Kant Vyas. Kamal Kant Vyas. His topic is to synthesis of the derivatives of thiazolidines and evaluation of their antimicrobial and antifungal activities. Kamal Kant Vyas.
Can I audible?
Yes, Kamal, you are audible.
Yes, I am sharing my screen. Ma'am. 1 minute, ma'am. Can I proceed, sir?
Yes.
Myself, Kamal Kant Vyas, research scholar, JNU, Jodhpur, Rajasthan. My topic is that to synthesis of derivatives of thiazolidines and evolution of their antimicrobial and antifungal activities. Next, abstract, a series of thiazolidines derivatives were synthesized for this investigation. Yield of 60 to 90% were obtained for all the synthesized compounds. NMR and IR spectrum analysis, as well as carbon, hydrogen, and nitrogen studies were used characterized these novel synthesized derivatives. All synthesized derivatives had analytical and spectral data that agreed completely with their postulated structure. Antibacterial and antifungal tests were conducted on all the newly synthesized compound. Antimicrobial activity is modified by the presence of heterocyclic nucleus. Ciprofloxacin and fluconazole were utilized as reference medications, antibacterial and antifungal research, respectively. The result of the screening showed that several of compounds had stronger antibacterial and antifungal properties than the standard medicines. The next, introduction. All heterocyclic compounds are great interest in pharmaceutical chemistry. Out of these heterocyclic compounds, the benzofused heterocyclic compounds, that is thiazolidines and its derivatives, have widely variety of biological activities. The synthesis of thiazolidines derivatives is topic of much research and discussion. The two carbon nitrogen bond in thiazolidines, when disconnected, give orthophenyldiamine and formic acid. Therefore, thiazolidines synthesized by condensation of diamine and carboxylic acid or carbonyl compound. Ammonium salt can be used as a catalyst. Transition metal catalyst, namely copper 2 acetate and lead tetraacetate, can be also used. Next is research methodology. It includes synthesis of differently substituted thiazolidines derivatives. This process involves two steps. First steps lead to development of three parent compound A, B, C. In second step, each parent compound was converted into four derivatives: A1, 4, B1, 4, C1, 4. In all, a total of 12 derivatives are synthesized. The required chemicals were obtained from a sigma Aldrich. The structures of the prepared derivatives were elucidated by the spectral analysis. All derivatives were evaluated from antimicrobial activity. And synthetic procedure method used in a 250 ml RBL skip base, 0.1 mole, 3.6 gram in benzene was taken. Dean Stark apparatus was attached to it, and thiological acid 0.01 mole, 0.92 gram in benzene was added slowly. Then it is refluxed for 15 to 16 hours. During the course of the reaction, the water was removed continuously, and the benzene was distilled off to get a thiazolidines, and the solid product was filtered, dried, and recrystallized from absolute alcohol. And synthesis, 3 amino acid named valine, leucine, and isoleucine were used. Synthesis of amino acid derivatives for thiazolidines. In first step, amino acid has been reacted with C base and from parent compound. In second step, parent compound reacted with the thioglycolic acid, which result in the formation of various thiazolidines derivatives. Next, it is the reaction part of the valine, using by valine scheme first, and second is used leucine, and the reaction is part is there, and third one is isoleucine used, and it is a physical characterization data of compound A1. You can see it is the chemical formula, extract mass, molecular weight, and M/Z ratio, elemental analysis, and melting point is here. Then physical characterization data of compound A2. It is a chemical formula, extract mass, molecular weight, elemental analysis, and melting point. And this is the third one, 3-methyl 2,2-nitro-4-oxo, 3,2-phenyl acid amido, 1,3-thiazolidine, 2-ill-amino butanoic acid is formed. Chemical formula, extract mass, and molecular weight is 396.42 M/Z ratio. Elemental analysis and melting point is 011.54 Kelvin. And it is a NMR spectra of A1 compound. You can see for A1. And second is NMR spectra for A2 compound. NMR spectra is here. And third is spectral data of compound A3. And it is the table for antifungal activity. And different compounds are here. A is study. And compound A1, A2, A3, A4, B1, B2, B3, B4, and C1, C2, C3, C4 are 12 compounds are here. And result and discussions. The primary focus of the study was synthesis of a number of substituted thiazolidines derivatives, scheme 1, 2, 3. Excellent yields of 60 to 90% were obtained for all the synthesized compound. Spectroscopic IR and H1 NMR approaches were used to verify the structure of all freshly synthesized derivative. All synthesized derivative had analytical and spectral data agreed completely with their postulated structure. Antibacterial and antifungal properties were tested for all synthesized compound. The inhibitory effects ranged from the mild to strong across all the substances. For what we can tell from the screening fields, several of the compounds have far stronger antibacterial and antifungal properties than the standard medications, sir. Some references are, and thank you, sir. Thanks.
Thank you, Kamalkant. The
Thank you,
session
ma'am.
is open for the questions.
Yes.
I have a question.
Yes, Shrutika.
Uh, Sir, can you tell me from like which amino acids the efficiency of derivative was better? Like you have mentioned that 60 to 90% recovery was done. Efficiency was 60 to 90%. But from which amino acid can you define it was better to form the derivative of your compound?
Ma'am, it is leucine, isoleucine, or valine amino acid is used for research, and its yield is 60 to 90%. It is a basic amino acid, so I used it.
Sir, like among leucine, isoleucine, and valine, which was better to form the derivative?
Which one?
Which was better?
Acha, leucine, ma'am.
Okay, ठीक है. Thank you.
Kamal Khan, just tell me that you have done antibacterial and antifungal studies also, right?
Yes, sir.
So why you are interested to do these activities? Do you know that your compound would be better as an antifungal or antibacterial, and just you have done, you synthesize and just you are checking just to add one more data? One question is that. Another one, which one is showing high grade of activity? Means which compound is very good antibacterial or antifungal? And
Sir,
what is the reason for that? Why they are showing that?
sir, antibacterial and antifungal compound of ciprofloxacin and fluconazole were utilized, sir, in medication, sir. It is a basic compound, sir.
So how they show these antibacterial and antifungal activity?
Sir, it is the, sir, basic medication, sir. So I used this type of
Okay, no problem. Okay, Kamal Khan.
thank you, sir.
You. So if anybody wish to ask, okay, then we can move to other candidate. Yeah, Deepthi, we
Yes,
can
sir.
switch over to another one.
Thank you, Kamal Khan. So the next participant is Harish Kumar Chaudhary. His research topic is your microcrystalline cellulose and itaconic acid-based pH-sensitive semi-interpenetrating network hydrogen for oral delivery of insulin. So Harish
Hi,
Kumar, you can present
yes,
your.
yes, sir. Yes, ma'am. Good evening to all. So this is the presentation that I'm going to do. So I have prepared this microcrystalline cellulose, the hydrogels of this microcrystalline cellulose and
Harish, your
itaconic acid.
presentation is not visible.
It's not visible. Okay. Is it visible?
Yes, yes.
Okay.
Please put it in the slideshow mode.
Okay. Okay.
Yes.
So this is the so I have prepared this pH-sensitive semi-interpenetrating network hydrogels for oral delivery of insulin of this microcrystalline cellulose and itaconic acid. So first of all, this cellulose this is just to introduce about the cellulose. So it was in 1838 that Emils Payne, while studying some different types of foods, he came to know that there is a knew that starch or glucose may be present, but this could not be broken down to basic units of glucose just as a starch. So he named that the new thing that cellulose, because he obtained this from the cell walls of the plants. So Payne isolated this it from the plant matter and determined its chemical formula. Later in 1870, Hayat manufacturing company used cellulose to produce the first successful thermoplastic polymer. In 1920, Hermann Schrödinger determined the polymer structure of this cellulose. And in 1992, Kobayashi soda and the compound was first chemically synthesized. Anyway, this was something to about the cellulose. Now this microcrystalline cellulose, that is one of my ingredient that I have used in the preparing of this hydrogels. This microcrystalline cellulose is a term used for wood pulp or texturizer, the anti-caking agent, fat substitute, emulsifier, extender, bulking agent, and most common form is used in vitamin supplements or tablets. So this microcrystalline cellulose range particle size ranges from micrometers to 100 micrometers and is successfully industrialized for decades. It has an advantage of having a high mechanical strength and easily accessible, relatively low cost, and serve as a good candidate for phosphoring agent. Uh, However, the large particle size of this microcrystalline cellulose restrict its homogeneous distribution in these polymer networks. So, reducing of the particle size of this microcrystalline cellulose, it will be highly cost. It will be having a high cost. Um, so, usually, this surface modification of these particles is done to facilitate this uneven is even a distribution to the key borders to maximize its reinforcement effect. So various types of forms of cellulose have been there, including that is been permitted by FDA, including a fat substitute, bulking agent, low calorie foods, texturizer, emulsifier, extender, and food products are obtained from this pulp and fibrous materials such as wood and cotton. Although it has a pharmaceutical application such as filler and tablets, it is they represented novel and more successful cellulose powder, mainly used in the pharmaceutical industry as a diluent and binder to tablets. It has an advantage over conventional cellulose due to its high specific surface area and due to its biodegradability. This microcrystalline cellulose is widely used in the pharmaceutical applications. In food industry as a thickener, fat substitute, dairy product, and stabilizer in the creams. And very recently, this MCC-based hydrogels have been used in the in vitro delivery of the flexing. So, so our topic being the oral delivery of insulin, we know that diabetes is one of a very important disease that has been a cause of a large amount of deaths worldwide. And this is associated with the malfunctioning of the pancreas to develop the sufficient insulin. Almost 30 years ago, therapeutic proteins such as insulin was used, and later on the US FDA approval was given to insulin to use as a major therapy for diabetes. However, the main problem lies with the delivery of these proteins and peptides. So far, subcutaneous route has delivered, but at the same time, it is often feared by the patients due to their prick, however small it may be. And in this context, the oral route has gained an importance. So oral route is simpler and economical and a first choice of patient for its comfort. And hydrogels for say associated with some problems associated with lies with the effective availability of the insulin due to the enzymatic degradation of this insulin in the body. And to ensure its availability, it is important to prevent enzymatic degradation. Human body is a delicate system. It could be several mechanisms to deal with a foreign agent introduced in the body by any route. A deeper insight of molecular biology and biochemistry is required for such applications of proteins and peptides. Of the several methods available for the drug delivery, hydrogels have gained importance due to their remarkable properties, such as ability to retain large amount of water, excellent biocompatibility, and their mechanical properties which suits the body. In 1960, Listerine and Lim, the hydrogels were successfully used for the contact lenses And these hydrogels, due to their novel properties and structure, can be tailored precisely to desired therapeutic delivery of the drug. And GI tract is a series. This is about the anatomy of the human body. GI tract being a hollow organs joined with twisted and tube with a mouth, the anus, and hollow organs being this mouth, esophagus, and stomach, small intestine, large intestine, and so on. These solid organs do not play any significant role in the oral delivery. So the protein and peptides that are used for oral delivery have to face some challenges. And challenges, uh, two main challenges are uh, proteins and peptides are denatured in the acidic pH of the stomach or by the digestive enzymes and therefore inactivated. The passage of the drugs from the epithelial membrane to the bloodstream is quite difficult due to their poor permeability. Hydrogels, owing to their three-dimensional polymeric network, consists of cross-linked hydrophilic components which have high water and biological fluid retention capacity, and while remaining insoluble. Hydrogels, by their complexing properties, can remain in stable and uneven conditions of stomach and protect the drug from the denaturation by acidic pH and digestive enzymes. And further, drug can be released in the small intestine, which has a lower population of enzymes, neutral pH, and larger uh, neutral absorption. So these here they are decomplexed in the upper intestine, as you see in this figure. So hello, am I audible, sir?
Yes.
Okay. So we see that due to the decomplexation of these hydrogels, the drugs is released in this. So my okay. The challenges in this oral delivery is that due to this acidic pH, its degradation takes place in the stomach, and
Harish,
infusing a
we have limited time, so just finish it.
sir, just sir, I'm just trying to conclude. It is only a few slides that are left. So I mean, we're coming to the main thing, just to pass through this. Okay, so another ingredient of this is the itaconic acid, and you see that it has a two carboxylic moieties in skeleton, and this is one of the important thing that it is responsible for its pH sensitive nature. So this itaconic acid is obtained from the distillation of citric acid and is produced by fermentation. So work done that I have done is I have tried this microcrystalline cellulose and itaconic acid in different proportions and different compositions and came out with some of the best swelling ratios that I could have in this distilled water was somewhere around 2,127%. And these swelling ratios were calculated for other solutions as well. And these are the cellulose, these are the hydrogens that were obtained, and they are disintegration, and they were optimized with a different amount of cross-linker and this initiator. It was made by free radical polymerization. Okay, coming to the samples that I prepared. So it is shown that this K12 sample is showing the best swelling ratio, and later on these ratios were calculated in distilled water and saline solution and glucose. It shows that lower amount of these swelling in the saline solution and even greater in the glucose, and the highest being the distilled water. And when their pH sensitive nature was also studied, and it was seen that the lower pH below the 7, that is up to 4, it remains almost in the collapse state. And as the pH of the solution increases, or the increases, it swells up, and during its swelling, it gives out whatever the things it has encapsulated in these hydrogels. So these in vitro analysis of these hydrogels were also done, and it is quite clear that these uh, these hydrogels, upon getting this insulin encapsulated, and they show remain in a collapse state at a very low pH of 1.2, and as the pH increases, they just swell up and release the
Harish,
insulin.
please come to the conclusion.
So these later on several methods were used for characterization, and these are the same, and these are there are references, and this is thanks for this. Thank you. Yes, sir.
Yeah, participants, if you wish, you may ask.
It is a request to all the participants, kindly adhere to the timings. The timing allotted for each presentation is maximum 10 minutes. So try to complete or wind up in 10 minutes. Any questions from the participant side?
So, Mr. Harish, what is
Yes, sir.
what is your conclusion technically?
Sir,
The compound, the polymer which you have prepared,
Yes, sir.
is it in better towards betterment, or it is still you are working to make it better?
No, sir. I have done all their studies, and in vivo studies has also been conducted, and their results have been produced. And it is found that these are very good and agents which can effectively deliver this insulin without having any. And moreover, the ingredients being the biodegradable, it does not have any other effect on other body. That their toxicity MTTSA also has been done. So they are excellent materials which can be used for this oral delivery of insulin.
Okay.
Yes, sir.
Okay, Harish. Thank you.
Okay.
Thank you, Harish. The next participant is Riya Singh. Riya Singh.
Can you hear me, ma'am?
Yes, her topic is computational investigation of triphenylamine based whole transport material for application in efficient perovskite solar cells. Riya Singh, you may start.
Yes, ma'am. Putting it to slideshow, one second. A very good evening to all. Myself, Riya, and I'm research scholar at Sharda University. My topic of presentation is computational investigation of triphenylamine-based whole transport material in efficient perovskite solar cell. Basically, if we look at the current demand of energy, it's increasing drastically. And with that respect, if we look at photovoltaic installation in the past 10, 13 years, it has been drastically increased, and we can see India participating as well with 6%. But of course, we want to increase this particular number. Currently, we are using silicon-based solar cell, and we are looking for the alternative as well, since it's costlier and not everyone is able to use that. With that respect, perovskite solar cell is one such alternative that we can use. And perovskite refers to a class of material with specific crystal structure, and it is named after a mineral that is calcium titanium oxide. Basically, it has formula as ABX3, where we have cationic form as A, B is smaller cationic form, and X is anionic form. Why perovskite is currently in limelight? It's because of its evolution, a very fast-paced evolution we can see in the third generation of PV technology. And we can use it for large-scale fabrication. It's very on flexible sheets. Other than that, it's ambipolar as well. If we talk about silicon, it's not ambipolar. Either it conduct hole or it conduct the electrons. But if we talk about this particular material, it is ambipolar as well. If we move to high charge carrier mobility, that is one such thing that we require whenever we talk about the semiconductors or these such devices. We want long diffusion length that's present in it. It has high open circuit voltage and low cost processing of fabrication is there. And if we look for the increase in efficiency, around when it was started, it was around 3-4%, and then it was increased, and up to 26% it is currently. The major, not everything is gold over there. Some things are there that still need to work upon. One such thing that is the issue with this particular material, it's degradation. So it degrade very, very fast. So the layers are very unstable as well. And currently they were using lead in it, which is very toxic. So we need replacement for that as well. And the reproduce, if we talk about being to upscaling it, we find a certain difficulty for that respect as well. So if we talk about the working of PSC as whole, how can we eliminate all these issues? Is with use of two more layer. So perovskite is the active layer, but we can use hole transport layer and ETL, that is electron transport layer, in this particular material to give it more stability. So the work is very simple to separate the hole and the electron whenever the light falls on the perovskite and they jump and the pair is formed. We want it to get separated, and a little time we require, after which it can recombine, but we want that time to separate them out. So I am working on the whole transport layer, and the main work is to extract the whole and transport it. It also acts as an energy barrier to prevent the transfer of electron. Also, it gives stability because now the main layer is not in direct contact with the air or the moisture. Also, if the homo is like well fitted, well matched, then of course it can improve the circuit voltage as well. So the ideal HTL we want to be dopant free. As I have said, the lead is the toxic one, so you want a replacement for that. We want it to be efficient, low cost and stable. And one such thing that are working currently in this HTM are some material. We mainly focus on organic, being simple and synthesizing. Spyro is the one in HTM that has the highest efficiency that has been achieved, around 1670. For Spyro, we are using it as an HTM. But the problem with Spyro is that very costlier. So we are looking for some other simple organic molecules like TPA, triphenylamine, carbazole. I am working on triphenylamine based HTM, and I have designed 5 such small molecules. So I have named them as, coded them as M1, M2, M3, M4, and M5. So basically the structure is a donor receptor donor style, and they have various core molecule like thiophene, BTT, that is benzo trithiophene, BT, and pi linkers are there, E dot, benzothiazole, and thiophene. Now, all the whatever calculation I have done is using a DFT, using a B3LWIP hybrid functional. So if we first look at the HOMO and the LUMO structure for these particular molecules, so here what we check is their distribution of charges. So we can see for M1 it is distributed nicely to the whole molecule, whereas for M2 we can see it is at one corner specifically, and for the LUMO it's at the center only. If we talk for M3, M4, M5, we can see M3 is comparatively better as compared to the other two. If we talk as a whole, I think M1 and M3, we focus more. They have the more accurate or the more distribution throughout the particular structure. Now, these M1 and M3, because the charge distribution are like fully over the full molecule, so it also give good charge distribution, gives also charge extraction can also be good. Recombination rate will be less in these two cases. Now, about their homolumo energy or their band gap we talk about. So using the two different set, I have calculated the band gap. Now, why two different set? One set is the more accurate or the more precise one. That is the 311. That set is more accurate. So the band gap calculated for all these, we can see that for 631G, all these band gap are narrower as compared to the other set that we have did the calculation for. And some of the material, they have the experimental values as well. So they were matching as well for M1 and M3. moving towards their distance. So the distance between the linker and the basically the donor and the receptor. So we look over here the symmetry. So the more the symmetry is there in the structure, the more it is able to pass the electron and hole as compared to the puckered structure. So puckered structure is not that much of use if we talk about. Basically both, like all of these are fine, but if we look for M2 and the M3, so there we can see a slight difference in the bond length. If we talk about tangle as well, M2, we can see a slight difference there, slight variation between the donor, two side of donor. I have also presented here the UV vis absorption for these molecules. If we look for the UV vis graph over here, the spectra over here, ideally the HEM should absorb within the 400 nanometer of range. So till 500 is also fine. Why? Because 550 to 900 nanometer is what the perovskite, the main layer, absorb there. So we don't want hindrance with that particular layer. So we can see that for M5, the absorbance is also high, as well as the range is also fine. For M1, it's fine as well. For M2, it's a little bit low. The absorption is little bit low, but it is lying in the right area where we want it to be. Talking about the dipole moment, the solubility of HTM again is very important because at the end we are going to fabricate it. So highly soluble HTM are preferred as they can be easily coated over the perovskite layer as thin film. So solubility is directly related to the dipole moment. So the higher the value of a dipole moment, the better will be the solubility for it. And we can infer from here that F4 will be the highest. So F4 has the highest of the dipole moment, giving it more chances to be good in the fabrication part. So easy will be the fabrication in this case. And at end I have done the MEP cloud. So basically to understand the effect of the position and orientation of the attachment of the ring on the core, the charge distribution is determined by plotting this MEP, molecular electrostatic potential. Now this is very important property, and this surface it define where the electron are rich, where the electron are not like deficit, and where there are no electron like neutral. So red part is representing electron rich, blue is deficit, and green is neutral. For M1 and M5, we can see electron rich part over there. So these two structure can be considered for further. Okay. So yes, if I conclude, so basically I have designed the HTMs with triphenylamine unit, and I have analyzed them with the Gaussian software, and the pi linker that I have changed, the properties have been observed that changing the linker has a change in the property, in their optical property. Now, if we talk about the charge distribution, we can see M1 is having good. Overall, all in all, we can see M1 is having most of the property that are well and fit. So that can be considered for further work of synthesizing it. And also I'm further analyzing it with other properties as well. So yes, that's it. Thank you so much for listening.
Thank you, Ria. Thank you.
Ma'am, I can't hear you. Can you speak a little bit louder?
Yes, thank you, Ria. Now I'm audible.
Yes, ma'am, audible.
The session is open for the questions. If any participant would like to question her. Hemant sir.
Yes, Deepti.
Yes, sir.
Ria, tell me which software you are using for these
Sir,
DFT calculations?
said for DFT calculation, I'm using Gaussian, but other than that, I'm using multi-WFN as well. And there is one as VMD. So these three currently I'm using.
So DFT, which version you are using?
So Gaussian 06.
Sorry, Gaussian, which version you are using?
Says sixth, sir. Basically, that is the one that I'm using currently. That is sixth of it.
Okay.
Gaussian view
So,
6.0, sir, if you know about.
So DFT calculation can be done for a like big molecules. Is
It
it
can be
easier,
done, not
or we have to do with the small molecules only?
if we can do so for big molecule also, it's just time consuming. So for big molecule, it will take more time. So like it can happen that it will take 1 week for a big molecule for one calculation.
Okay, so you are doing technically theoretical study, right?
Sir, after doing the theoretical, I'll move to the
hands-on.
synthesized part. Basically, I'll decide like which molecule is having good property so that I can compare it when I synthesize it.
Okay. Okay, Ria.
Okay, thank you,
Thank
sir.
you.
Thank you, ma'am.
Thank you, Ria. So the next
Okay,
participant is
Dipti.
Sakshi Pandey. Her topic is zinc, terephthalic acid, and copper curcumin nanoscale metal organic framework. Sakshi, are you able to hear me?
Hello.
Is Sakshi here? OB97. I can't find Sakshi. So we shall be moving to the next participant, that is Viral Kumar Arvind Bhai Doshi. Viral Kumar Doshi. So his topic is synthesis and biological evolution, 5-chlorothiophene containing novel hydrazide hydrazone derivatives. Viral Kumar Doshi, please be ready with your presentation.
Hello, ma'am. Am I audible?
Yes. So where did you go? We called you.
Ma'am, it's a network issue, I think.
Okay. So Sakshi, chalo start karo apni presentation. Viral, we will be having the presentation after Sakshi's
Yes,
presentation.
please, please bind up in exact 8 minutes.
Okay, sir.
Hello.
Viral, please make your presentation after Sakshi finishes.
Am I audible, ma'am?
Yes, you are audible.
Okay.
So you will speak after Sakshi.
No,
Actually, sir, we are
I
a
think
team.
they are a team of 2 students, sir. Sakshi and Aniket.
Yes, ma'am.
Okay.
Okay, fine.
Okay.
So may we begin, sir?
Yes, please.
Right. Good evening, everyone. Respected sir, I Aniket, along with
I, Sakshi.
undergraduate students of Rajdhani College, University of Delhi, are here to present our work on zinc terephthalic acid and copper curcumin nanoscale metal organic framework. So, first, what are nanoscale metal organic frameworks, or NMOF? So NMOF is a crystalline material composed of metal ions coordinated with organic ligands on nanoscale. These materials have high porous structure with nano-sized cavities which can be precisely tuned and controlled, making it highly applicable in different fields. They exhibit a wide range of properties, including gas storage, catalysis, drug delivery, and sensing. Next, why did we choose the zinc terephthalic acid system? It is because of the following uses. First one being catalysis. Its large surface area and tunable pore size make it suitable for catalytic processes. Also, it has great uses in drug delivery. That is, drugs can be encapsulated within the pores and released gradually, improving the therapeutic efficacy. Also, they can be used in sensing. That is, it can be functionalized to detect specific molecules, making it useful in chemical and biological sensing applications. Also, it can be used in energy storage. It has a potential application in energy storing devices such as batteries and supercapacitors. These are some of the few uses that they have. Next, this is the structure of the zinc terephthalic acid system. Now, I would like to request Sakshi to explain the further synthesis procedure.
The procedure is as follows. First, the NMOF zinc terephthalic acid system was prepared using micelles formation by the help of commercial surfactant sodium dioctyl sulfosuccinate, which is also called AOT. The solutions of zinc acetate and terephthalic acid were prepared in dimethyl formamide. Second, the reaction mixture was first stirred at 75 degrees Celsius for 12 hours, and then after addition of ethanol, it was centrifuged to obtain the product. This was washed with ethanol and it was vacuum dried to obtain the final product. Now, next is the copper curcumin system. We choose the system because of the following uses. Firstly, gas separation. Due to its tunable porosity, they are suitable for separating gases such as hydrogen and carbon dioxide. Second, antibacterial agents. Copper ions within the MOF structure have inherent antibacterial and antifungal properties. Third one is photocatalysis. It can harness light energy to drive chemical reactions, which is valuable in processes like water purification and environmental remediations. Next is the possible copper curcumin structures. Then next is the procedure for the synthesis of copper curcumin system. This is same as the zinc terephthalic acid system, but we have used copper acetate in place of zinc acetate and curcumin in the place of terephthalic acid. Now, here are some glimpses of the procedure we followed. Then this is the characterization. The confirmation for the formation of NMOFs was done using IR report. Now, I would like to ask Aniket to explain applications and future aspects of our work.
Now, the applications for these nanoscale metal organic framework are as follows. First one being biomedical imaging, that is, MOFs can be used in imaging techniques such as MRI. They can also be used for gas purification, that is, they can capture impurities from industrial gases and contributing to cleaner and more efficient processes. Also, they have applications in nanomedicines, that is, they can be utilized in targeted therapies for cancer treatment, delivering drugs directly to tumor sites. Of course, the whole procedure was not so smooth sailing. The nanoscale comes with its fair share of challenges. The prominent one being stability. That is, metal organic frameworks can be sensitive to moisture or high temperatures, which limit their practical applications. Also, scalability. That is, synthesis of these on a large scale can be challenging and really expensive. We also have to consider its toxicity. That is, some of the components may be toxic and their safety in the use of medicines need to be carefully considered. Also, the selectivity. Achieving high selectivity for specific molecules or ions in gas separation or catalytic processes can be challenging. Of course, these challenges may have been, but apart from these, they have some really good future aspects or future applications. The prominent one being flexible electronics, that is, MOFs with electrical conductivity properties can be incorporated into electronics, opening up new possibilities in variable technologies and flexible displays. Also, they can be used in space explorations, that is, for gas storage, for long duration of times, for longer missions, or for life support systems. They can also be used for...
We have a limited time, so you have to finish within a minute, please.
Okay, sure, sir. Also, they have smart material applications, that is, due to their tunable properties, they can lead to the development of smart materials that respond to external stimuli. In conclusion, we got to learn many applications of the metal organic frameworks. Of course, there are many challenges in the process of their synthesis, like long-term stability and selectivity. But regardless of all the challenges, it is a very new and ambitious field with a lot of future potentials. Thank you.
Audience, do you have any question? Sakshi, where you did this research? In college or?
That's at the Ishbandhu College, University of Delhi.
Okay. So, and what type of analysis you have done?
So the project was under Pratibha ma'am. We were actually supposed to prepare nanoscale particles, but of course there were certain limitations. So we decided to do nanoscale metal organic frameworks. And the systems that worked best for the time limit that we have were zinc terephthalic acid and copper curcumin. Of course, there were some trial and errors, so we tried some different.
No, my question is, what type of characterization you have done?
So we have done IR characterization, and we are going to submit for UV spectroscopy and NMR.
Okay, this is a preliminary research, you can say, right?
Yes, sir.
Okay. Okay, Sakshi. Thank you.
Okay, thank you, sir.
Thank you, sir.
Thank you. Dipti, we can move to the next. Viral Kumar.
The next participant is Viral Kumar. His topic is synthesis and biological evolution, 5-chlorothiophene containing novel hydroxyl hydrazone derivatives. Viral Kumar, please present your work. Everybody is requested to kindly finish your presentation in maximum of 8 minutes. We are already short of time. Viral Kumar. Viral Kumar, are you there? Kindly mark your presence. Are you there? You are unmuted, but you are not audible.
Move to next one, please.
So the next one is Saurabh Kumar. His topic is extraction of caffeine and sodium benzoate from different brands of cold drink available in the market. Saurabh Kumar.
Yes, ma'am.
Yes, Saurabh, present your work. Start your presentation.
Visual.
No, Saurabh, it's not visible. Yes, now you are visible. Now you are visible.
Good evening. This is Saurabh Kumar, pursuing BSc Honors in Chemistry, third year from Ramjas College, University of Delhi. I am here to present my work that I and Priyanka Yadav, under the guidance of our professor Dr. Savita Bargujar, did. Our work was to extract caffeine and sodium benzoate from different brands of cold drink that are available in market. First of all, why we chose this topic? We chose this topic because cold drink is a common beverage that is very much popular among school and college going students. But it poses certain problem because it contains certain chemicals and preservative that are harmful to us if we consume it in long term. So the main purpose of the project was to create awareness among students that these drinks are not good for our health if we consume it in a long term. Caffeine, we extracted caffeine and sodium benzoate via simple methodology of liquid extraction, and their qualitative analysis was done. For caffeine, we did murex test, and for the sodium benzoate, FeCl3 test was done. The most important part of the work was the quantitative analysis of sodium benzoate, means what percentage of sodium benzoate present in different brands of cold drink. The principle as different brands of cold drink that caffeine, as we know, it is an alcohol. It is an alkaloid. It is an alkaloid. It is an alkaloid. It has a psychoactive effect on our mind. It increases alertness. It improve attention in studies. That is why it is very much popular among the students. So that popular among students. Whereas sodium benzoate is a preservative that is very much common in cold drinks. It has two health concerns associated with it. Like when it comes with contact with vitamin C, that it produces benzene, and benzene, as we know, that is carcinogenic. Therefore, for the extraction purpose, the principle involved was liquid extraction. As we know that most of the soft drinks, all the cold drinks, the main content of these cold drinks is water. So we chose chloroform as a extracting solvent, in which caffeine is more soluble than the water. For this, 100 ml of cold drinks were taken, and 2 grams of sodium bicarbonate was added to it. It was stirred for 2 minutes, and it was poured into a separating funnel. Then it was shaken and allowed to stand. Then chloroform was layer being denser was taken out from the separating funnel. Then it was steam distilled off to get sample that was yellowish white residue. To confirm this yellowish white residue as a caffeine, we did the murexite test. For this, potassium chlorate was added to the brown bottom flask containing caffeine. Then few drops of dilute hydrochloric acid was added. It was heated and dried. Then ammonium hydroxide was added to it. Change in color to pink. That is because of the formation of ammonium purpurate. Proves that the extracted one was caffeine. It was observed that Coca-Cola Rave Sting contained caffeine in it, while there was no presence of caffeine found in Sprite. For the sodium benzoate, same extraction of liquid extraction process was used, but the problem was that sodium benzoate, being a salt, is more soluble in water as compared to chloroform. So first it was needed to convert it into benzoic acid so that it can be extracted from the water or water present in a cold drink. So for this purpose, first sodium benzoate was converted into benzoic acid, and then it was extracted by the same procedure. And further, the FeCl3 test was done, and change in color to brown confirmed that the extracted material was sodium benzoate, and it was due to the formation of sodium ferric benzoate, that brown color. It was means Coca-Cola didn't contain any sodium benzoate, while Sprite, Limca, Rave, Sting contained sodium benzoate in it. The most important part was the quantitative analysis of sodium benzoate that we did in the lab, and that via simple method of acid alkalimetry titration. For these two process there was two steps. First was a standardization of NaOH with oxalic acid, as NaOH is not a primary standard substance. Then determination of sodium.
Saurabh, complete within 1 minute, please.
Okay. So, determination of sodium benzoate was done via simple method of acid alkalimetry titration using phenolphthalein as an indicator, and some of the results after calculation, it was found that Sting contained highest percent of sodium benzoate, that is 2.33%, while Rave contained the least amount of sodium benzoate. Overall conclusion of this project was that Sting, being both containing highest percent of sodium benzoate as well as caffeine, is not good for the health if we consume in a long term. Especially it is said that one should not take 100 milligram of a caffeine, especially for the children of age group of 12 to 18. That is equivalent to 2 cans of soda, because it is going to cause a lot of harm to our body. Even the physical development will be inhibited by this long-term consumption of the such drinks. And according to Food and Drug Administration, permitted allowable concentration of that sodium benzoate is 0.1%. If a percentage exceed it, it will cause harm to our body. And such drinks are not, if even a single drop of such drinks is like a poison for the children of below 12 years of age. For this, I would like to thank our mentor, that is Dr. Sarita Bargulkar, and our principal, sir. Thank you, sir.
Any question from the participants?
Hello.
Yes.
Yes, please ask.
Yes, myself Yudhishthir. I want to ask a question. Two questions. Hello.
Yeah, please ask.
My first question is that.
Yudhishthir, you are muted.
My first question is that why you choose only beverage or cold drinks? Why you did not choose some tea leaves and coffee leaves? And the second question is that whenever we study about solvent extraction method, then we use separation or distillation coefficient. How about the distribution coefficient? What is distribution coefficient?
First of all, I chose cold drinks because this is a common thing that is being consumed in university area. That is especially where I am living. It is very common among the students, especially among the school going children, because school going children are not taking tea or coffees, but they are consuming these drinks. Like Sting is very much popular. That's why I chose. And about the distribution coefficient, means liquid extraction process is based on the principle that a solute and an light can distribute itself on the basis of solubility into two different immiscible solvent in a certain ratio, and that certain ratio is known as distribution coefficient for that particular.
Thanks, that's very good.
Okay, Saurabh. Thank you.
Thank you, sir.
Okay, Dipti, move to the next one.
So the next is Shrutika Singla. Her topic is determination and quantification of a 4 anticonvulsant drugs from urine utilizing DLL-ME, LC-MS-MS in forensic cases. Shrutika, you may start.
Good evening to all the delegates present here. I am Shrutika Singla, research scholar at MIT Institute of Forensic Sciences, MIT University, Noida. So today I'll be sharing my research topic, which is determination and quantification of 4 anticonvulsant drugs from urine utilizing DLL-ME, LC-MS-MS in forensic cases. Basically, the epilepsy is a non-communicable chronic neurological disorder affecting brain activities. It is characterized by 2 or more continuous seizures within 24 hours. It has become fourth most common neurological disorder and has been ranked as second most burdensome neurologic disorder by WHO's 2010 Global Burden of Disease. The rate of drug-facilitated crimes are increasing day by day, involving many categories of drugs, which include anti-anxiety drugs, day trip drugs, antidepressants, and even anticonvulsant drugs. These drugs are involved in self-toxication cases as well as multi-drug poisoning cases. It has been found to be involved. Some of the common examples of these drugs are levetiracetam, gabapentin, topiramate, valproic acid. Certain benzodiazepines are also comes under in this category. In this research, we have selected 4 drugs, lamotrigine, valproic acid, topiramate, and carbamazepine, based on their occurrence in different poisoning cases. We have applied dispersive liquid-liquid microextraction technique. Basically, my research focused on extraction, development of extraction protocol for these anticonvulsant drugs. So we have used dispersive liquid-liquid microextraction. This method is basically modified liquid-liquid extraction, and this method can be applied to extract small quantity of sample from biological matrices. In this method, 2 different types of solvents are used, disperser solvent and extraction solvent. These solvents are added to biological matrix, which results in formation of cloudy solution. In this study, we have used 1 milliliter acetone as disperser solvent and 4 microliter chloroform as our extraction solvent. The cloudy solution that is obtained was centrifuged at 6,000 RPM for 6 minutes, which resulted in settlement of organic phase. This settlement was then taken for LC-MS-MS analysis. The condition of LC-MS-MS was given in here. We have used 2 mobile phases. Mobile phase A was 5 millimolar ammonium formate in 0.1% formic acid in water, and our mobile phase B was 0.1% formic acid in 100% methanol. The concentrations of standards that we have taken were 5, 10, 20, 50, 100, and 200 ppb. And these are the chromatograms of LC-MS-MS that we have obtained after the extraction protocol. So our A, I have shown these in 200 ppb. The A was valproic. Our A was valproic acid, which is at 7.461. Our B is oxcarbazepine, which is at 9.028. 3 is lamotrigine, which is at 10.003, and 4 is topiramate, which is at 11.995 retention time. These retention times are basically in minutes. This is the linearity plot for all the 4 drugs that we have used in this study. And these are the LODs and LEQ that we have obtained. We have obtained less LOD for lamotrigine and valproic acid and oxcarbazepine and topiramate as well. The regression analysis that we coefficient of regression was under 1, which was under the validation range by ICH guidelines. For this study, we have followed ICH guidelines. So in the conclusion, I want to conclude by saying that we have developed an efficient and advanced DLLME extraction protocol for simultaneous extraction of 4 anticonvulsant drugs, which were oxcarbazepine, topiramate, lamotrigine, and valproic acid. This method is optimized and validated by ICH guidelines, and the LODs and LOQ obtained for the different drugs has been mentioned here. The LODs and LOQs obtained for these drugs were minimum as compared to other method that has already been published. The developed method can be used in forensic science laboratory for toxicological analysis in urine sample, as we have taken urine sample for our analysis. So these are some of the references for my study. Thank you.
Thank you, Shrutika. Any questions? Hemant sir?
Yes, participants, you may ask. Dipti?
Yes, sir. Yes, sir.
Your voice is not. Yeah.
Yes. Hanji,
Am I
sir.
audible?
Yes, sir. Yes, sir.
So, any question from the audience side?
So no questions being asked from the audience.
So, Ritika Singla, just tell me, you are in which year of the research?
So basically, I am in my second year.
Second year.
Yes.
So, what techniques you are using to analyze the data?
Sir, we are using different softwares. We are using Excel. Basically, we are doing regression analysis as well as ANOVA is being performed. We are using Stat Graphics and GraphPad Prisma.
Okay. So, it is an open access journal or you purchased it? Means your supervisor?
Sir, these are open softwares that are available.
Okay, okay. So, will you verify experimentally also some results?
Sir, these are experimentally verified and validated by ICH guidelines.
Okay. Okay. Singla, thank you.
Thank you, sir.
Thank you very much. Dipti, you can move to the another one.
Yes, sir. So the next participant is Yokeshwari. Her topic is modeling of electroreduction of halogen oxoanions via autocatalytic redox meditation by halide anions. You may start presenting. Yokeshwari.
Ma'am, are you audible? May I am audible?
Yes, you are audible.
Yes, I am. My topic is modeling of electroreduction of halogen anions, autocatalyst redox mediation by halide anions. Myself is G. Yogeshwari. I'm a research scholar under the guidance of Dr. L. Rajendran sir, Department of Mathematics, Amit University. Introduction. Mathematical modeling. Mathematical modeling is the process of converting real-life issues from one application field into structural mathematical formulation. A model aids in the explanation of system, the study of the impact of various components and the prediction of behaviors. Dynamic systems, statistics to different equations, and game theoretical models are only a few examples of mathematical model. Can be analyzed theoretical and numerical to provide solutions and important guidance to the original application. It is critical in a variety of applications and works well in a variety of other situations. Give problem solving solution, precision, directions. It also allows for a comprehensive grasp of the system model. It's a numeric and analytical solution. How to find the numeric and analytical solution? I am using the Maple software for analytical solution and using MATLAB software for numerical solution. There are so many methods to solve a problem by analytical method. It's a Paddy approximation method, Akbari-Conjay method, homotonic permutation method. So many methods by here using the AGM method, that is the Akbari-Conjay method. Introduction: electrode reactions and chemical reactions frequently coexist. The EC mechanisms which transfer the results of the redox transformation into a specific final electronegative product by an irreversible chemical process is one of the most frequent electrode reactions. This mechanism has been the subject of countless theoretical and experimental studies over the past 7 decades. The reactant and the product of redox couple adaptation also act as a coupling agent in most electrode reactions in aqueous medium. These problems of fuel cells have led to develop a system which are called redox followed batteries. They are RFBs. In the initial variant, both the analytical and cathode reactions present redox process which provide quasi-reversibly at cheap electrode materials inside the discharge cells where the electricity is generated while the product of both reactions are regenerated outside the cells, either chemical or electrochemically. I am using the Akbari-Conjay method. Akbari-Conjay method has applied to solve many non-linear problems in physical and chemical science. Akbari and Conjay developed the Akbari-Conjay method in year 2014. Can be considered a hyperbolic function, semi-analytical approaches for solving strongly non-linear equations with boundary problems in finite domains. AGM is easier to apply compared with other semi-analytical techniques. It's a solving process such that only the initial boundary conditions, the main differential equation, and the derivatives are required. This is my model of halogen molecules, halide anions, and halogen ions. That is equation 1 and 2. The boundary conditions are u dash of x is equal to minus v dash of x, and w dash of x is equal to 0 at x is equal to 0. U of x is equal to 1, v of x is equal to 0, w of x is equal to 1 at x is equal to 1. This is the equation 5. This is the solution. 6, 7, 8 is the solution of halide anions, legends, and using AGM method, where L and M are obtained as follows. This is my numerical analytical compare graph for U of x, V of x, W of x. That is a halide anions. When A equal to 0.8, L equal to 0.5, and M equal to minus 0.7, and B when A equal to 0.8, L equal to 0.25, and M equal to minus 0.350. This is another graph for comparing numerical and analytical solution. Conclusion: the nonlinear second-order differential equation system is a new autocatalyst mechanism in solving using the AGM method. This method has been established for studying an electrode reaction coupled with halide homogeneous chemical reaction. Analytical result for the concentration are compared with numerical result. A satisfactory arguments observed between the obtained analytical results and the numerical results for all values of parameters. A satisfactory argument is analytical result for various parameters. These are my reference. Thank you for the opportunity.
Yes, participants,
Any question? Yokeshwari, what
Yes,
will
sir.
happen if you will choose?
So I'm not audible, sir.
What will happen?
Sir, your voice was
Sir,
not audible.
sir, your voice is breaking, sir.
What result would you expect if you will use non-halogens? Then, like oxygen, sulfur, if you will use, then
So, I'm, sir, your voice is not audible, sir. Breaking your voice, sir.
okay. So anybody, if you wish, then you can ask. Otherwise, we can go for the next one.
Okay, thank you, Yokeshwari. Our next presenter is Aruna Milan Sudam. The topic is chromium removal from water by activated carbon developed from waste rubber tires. Aruna,
Yes,
you
ma'am.
may yes
एकदम साइड में रुको जहाँ से
you may start your presentation. Your PPT is not visible, Aruna.
Yes, ma'am. I'm sharing
Okay.
right now, ma'am.
Yeah, it's visible now.
Good evening, all. I am Professor Aruna Sudame. I'm associate professor at ICHE-2023 College of Engineering, Nagpur, as well as a research scholar. And I'm going to present on my topic, research topic, chromium removal from waste from water by activated carbon developed from the waste rubber tire. So this is the introduction. I'm just telling about some facts about the waste tire. Actually, the production of tire increases due to automobile industries because it increases globally, not only in India, but it increases globally. Nearly 1 billion tires are disposed each year. More than 2.5 million tons of scrap tires are disposed every year, and more than 50% are discarded without any treatment. And by the year 2030, the number of waste tires would reach around 1.2 billion. And this is the actually the problem for the environment. And not only environment, all types of pollution can be increased with this, maybe air pollution, maybe water pollution, all types of pollution which can be increased by increasing the waste tire as a scrap. So discarded tires are one of the adsorbents that can be conveniently used for the removal of heavy metals. Actually, this is the waste tire. It can be used as a adsorbent, and it's a very good adsorbent because there are the number of some adsorption methods. But this adsorbent, it may be the definitely it's cheaply available, and that the costing definitely it's less. And another solution for the waste, that the waste tire which will be provide or increases the waste, and that the waste can be decreased by using waste tire as the adsorbent. Now here that is a study which is going, and the chromium 6 which can be taken as an element, and the carried out the experimentation is done around 100 ppm and 50 ppm concentration of chromium which is prepared, and the experiment is done with the 0.5% and 1% weight of adsorbent. Adsorption efficiently with respect to time is evaluated. The all 2 to 3 parameter which is evaluated, that is one is the adsorption efficiency with time, and another is the pH. Now this is a result which is got with the help of that is OES, that is optical emission spectrophotometer, and this is a standard that is a graph which is obtained here. We can see Now these are the results we got uh, for that is 100%, and we can see here that is a comparison 0.5% and the 1% weight. So here we can see that is around for 1% percentage of weight, we can see here that is a percentage absorption capacity is a 60.5. Now actually after this, once I got this result, so after I did another activation of this activated carbon with KOH again with impregnation, and I got a that is very good result, and that result is around 80% absorption capacity. So this is a graph. Here we can see that is how the absorption can be seen here. Now the again that is with the 50%. So I already took the different concentrations are there. So with the 50% results is very good. Here we can see that is around 1%, 50 ppm with the 1% weight. We can see here the 72.8% result is there. So in this way, and that another is a 100 ppm again 100 ppm. So we can hear that is around 60% by the 1%. That is about the concentration. In initial one there is a pH. So in the pH we can see that is a 84.4%, and for the concentration we can see here the 60%, and at 60% that the absorption capacity will be that is it will not be changed, and that will be the only the 60% we can see. So from the summary, or we can see here that this activated carbon, which is got from the waste tire, that can be, it's a very good solution for the adsorbent. It can act as a good adsorbent, and at the same time, we can, and again, because I didn't see here the result for the activation, but the activation I did, and after activation, I got a very improved result, and that will be goes more than 80%. So this is my work is there. These are the references. And thank you so much for giving me an opportunity to present my work with you. So thank you so much.
Thank you, ma'am. So any questions from the audience? Hemant sir, you may ask.
Yeah, Miss Aruna, chromium 6 is having
Toxic
some
than the chromium 3. Yes.
yeah, harmful effect, right?
Yes, yes, sir. Yes.
So why it is so? This oxidation state is showing harmful effects.
Because it have that is a 3 is have that is a less oxidation, na? And that the 6 is have a more oxidation. It's undergoing more oxidation fast.
Yeah, the chromium 6, why it is hazardous in nature as compared to chromium 3, right? Normally there are studies that we convert chromium 6 into chromium 3, which is less hazardous, right? So there is something else with the chromium 6. That's why it is hazardous or more dangerous for the environment. So have you done any literature survey in this regard?
Yes, sir. I have already done the literature survey. Yes. So that is the oxidation is that is very much that is a high that is a very fast. That is the very first reason.
Okay.
That is the very first reason which is undergoing oxidation very fast, because those metals which are undergoing the oxidation fast, so that's why they are actually they are going undergoing the process of what corrosion I can say, or it will be going to be the brittleness fast, and that's why the having much more effect, immediate effect on the metals particularly. And when it's showing the metal effect on the effect, so it will be showing the effect on the water as well as in the environment.
So you are using only one method to characterize it, right? OES, I think.
We are using, sir, both the methods. We are using, this is the optical emission method, and another that is a titration method that which is carried out in the laboratory. And then we are compared with both the methods are there. So almost in both the methods we got the much more similar result. Actually, first of all, we have to go to the titration method.
That's fine, Aruna. That's fine, no problem.
Yeah, sir. Yeah.
Okay, okay, thank you.
Okay, thank you, sir. Thank you so much.
Thank you, Aruna ma'am.
Thank
So
you,
the
ma'am.
next participant is Khemraj Meena. Khemraj Meena, you may kindly present your work.
Yes, ma'am. Am I audible?
Yes. Aruna ma'am, please stop your presentation.
Yes, ma'am.
Ma'am, slides are visible.
Yes, your slides are visible.
Okay, first of all, thank you so much, ma'am, and sir, to giving this opportunity because my presentation was scheduled yesterday. But
Khemraj, it is just showing screen sharing, but no slide is there. Please do it again.
yes, ma'am, just wait. Now is it visible, ma'am?
Yes, now it is visible.
Yeah.
Kindly put it on the, okay, that's all right. Yes, you may start.
Yes, yes, yeah. Good evening, ma'am and sir. Today's presentation, my topic is on lipopeptide antibiotics, and this presentation was scheduled yesterday, but due to the technical issue and internet connection, this was disconnected, and now this is scheduled. And I thank you so much for giving this opportunity to present today. My topic is lipopeptide antibiotics, and we see that in the current scenario, there are a lot of problem due to the chemical surfactant, because chemical surfactant in the nature, which are very hazardous, and chemical surfactant hazardous as well as that pollute the environment and pollute the water pollution, and also cause many type of the health risk for the human. So in our study, we tested the lipopeptide molecule, which are the antimicrobial compounds, the source of the special type of bacteria. Yes, sir. Hello.
Yes, you can go ahead.
Yes, yes. Yes. Yeah. And the special lipopeptide molecule, which have several advantage over the chemical surfactant, because these molecules are the non-toxic to the plant and animals, and these molecules are high, have biodegradability, eco-friendly, compatible, and no irritancy. Among the antimicrobial compound, cyclic lipopeptide molecule, which have especially surfactant, iturin, and fungicin, which these three type of the molecule which has important role, and which have you can say that versatile weapon, because due to their wider applications. These molecules are low molecular bioactive molecules, and we can say that molecular weight is a maximum 1 to 2 kd molecule, and you can see that a very less molecular weight, therefore they can easily penetrate in the membrane. And generally, if we see the source of the bacteria, especially microorganism, so Bacillus genus, which is considered as a microbial factories for the production of these type of the molecule. Bacillus genus, which devoted 4 to 5% of genome for the synthesis of the iturin, surfactant, and fungicin, the lipopeptide molecule. And these are the molecule, especially iturin and fungicin, we can surfactant, we can see that both molecules are the heptapeptide. Heptapeptide, which contain some amino acid, D amino acid, and some amino acid are the L amino acid. And this is amino acid makes the cyclic molecule, and which attach with the 12 to 14 a hydrocarbon chain, which gave a particular strength to the molecule, because without the hydrocarbon chain, this is very sensitive to the proteolytic enzymes, And after adding this hydrocarbon chain, it is more stable, thermostable as well as proteolytic enzymes. We know generally that lipopeptide molecules, protein molecule are generally synthesized from the translation process. Means there is a requirement of the ribosome. Without ribosome, translation is incomplete. There is no possibility of synthesis protein. But these molecules are synthesized with a special enzyme which is called a non-ribosomal peptide synthetase. This enzyme is a module of different unit and different domain that contain a different units which synthesize a particular amino acid as per the requirement of the lipopeptide molecule. Here we can see that there are a different domain for mycosubtilin operon, surfactant operon, and plipastatin operon. In our work, what we did, first of all, from the bacteria, especially from Bacillus subtilis, we isolated lipopeptide by the APME and BEMS method. After the extraction, we found that by the Bradford assay, we found the protein quantification and lipid content by the Sudan 4 test. After that, we conducted antifungal activity with the well diffusion method. And we found that this molecule, the whatever molecule lipopeptide we extracted from the Bacillus subtilis, it was very active. And in this picture, we can see that after the third, fourth, fifth day, and last at the fifth day, mucor and aspergillus both are the fungus strain which cause a particular disease in the especially in plant. And mucor, if we talk on the mucor, this mucor species it causes a disease especially in the capsicum, and this completely destroy the plant. And 77.8% and 52.4% activity of the both fungus strain was inhibited in the well diffusion test by the using this lipopeptide active molecule. After this molecule be purified by the different techniques. First, we collected cell-free extract. After the first of all, the bacterial cell was grown in the LB broth, and after the centrifuge and the cell-free broth means supernatant we collected. After supernatant, we added different concentration of the ammonium sulfate. Ammonium sulfate, what is the role of ammonium sulfate? We can say that ammonium sulfate purpose of this adding the ammonium sulfate to purify the molecule means precipitate the molecule. This ammonium sulfate work on the salting out process where the ammonium sulfate interact with the protein, and at the higher concentration of ammonium sulfate, protein interaction occur and precipitate in the medium in the tube. So visually that is the initial step of the separation. After that we go for the dialysis.
Please finish within a minute.
Yes, sir. Yes. Dialysis is the technique of the removal, salt removal technique. After that, the DE cellulose on which on the ion exchange chromatography on the basis of the charge separated, this molecule was separated finally on the size exclusion chromatography. This is the table of the purification steps where we can see that the specific activity was enhanced and the purification fold was also enhanced by using the different purification techniques. And a UV visible spectrophotometer on the basis of surfactant iturin and surfactant both we standard use. And on the upper C is we can see that is the purified lipopeptide that match similar to the surfactant. And in the thin layer chromatography, the white spot show the presence of the lipid, and the B this spot green spot like that present the amino acid that show the compound is the lipopeptide. Here we can see the this molecule lipopeptide is very stable at the high temperature. If we go for the autoclave at 100 degree centigrade, it was the active at 100 degree, even 120 degree centigrade. So we can see that it is also stable and it was active after 100 degree centigrade. And this was the test for we conducted in the in pot experiment where we conducted that this is the capsicum plant. ABC, there is a healthy plant, and DEF. These are the tested and control. Control plant was the very you can say that only fungus inoculum was there, and tested fungus with the lipopeptide. So we can see that in the tested plant were healthy, while control plant was completely died, or you can say that it destroyed with the infection of the mucor in the root. This is the structure in the antifungal activity is the without treatment of the lipopeptide fungus, and B is the with the incorporation of the lipopeptide. You can see that there is a fuzziness structure of the mycelia that show that it disturbs the integrity stability of the mycelia. There is a MIC value.
Cameras,
This is
please conclude it.
yes, just 1 minute, sir. And this is the antibacterial activity. Anterior activity after except the Shigella flexneri, these lipopeptide molecule inhibit the growth of the E. coli, Aspergillus, and Pseudomonas, and Pseudomonas, and Klebsiella pneumonia, and Salmonella typhi. So conclusion is that it was the molecule which was very, very active against the phytopathogens as well as in the bacterial pathogens. And this molecule was found very thermostable and active at 100 degree. That so that it was very active, and if molecule used, then at higher temperature also it can be used for the control the different type of pathogen like bacteria as well as fungus. Thank you, sir. Yes.
Yes, any question? Okay. So, Mr. Cameras,
yes, sir.
why you have chosen these compounds to study antifungal and antibacterial, especially this antifungal one?
Yeah, sir, because this is the work of my PhD, and I was in Simla, and I was working on the lipopeptide, and where capsicum disease is very, very prevalent. So then I focused the capsicum disease, especially that caused by mucor, and I targeted mucor fungus by this lipopeptide that is very, very thermostable, and it was also effective against, especially against the fungus mucor. That's why I chose.
Okay, right. Okay, thank you, Cameras.
Thank you so much, sir. Thank you so much,
Yeah.
ma'am.
Dipti,
Yes, sir.
we can move
So
to
we
the
are
last
left
one.
with the last participant, Viral Kumar. Viral Kumar,
Yes, ma'am.
kindly start
Just
your
sharing my.
Kemraj, kindly stop sharing your PPT.
Yes, ma'am. Yes.
What happened?
Hello.
Viral Kumar, your PPT is not visible.
Yeah, just ma'am, I have shared it, but I don't know why it is.
So if you are not able to do this, so then we have to end it here. So just try once more.
Yeah, just a minute, sir.
Yes, Toshi, nothing is coming out on the screen. What happened? Is there any technical issue from your side?
So Deepthi, I think he's having some technical problem.
Yes,
So
sir.
can you adjust him tomorrow if you have some
I think,
presentation?
sir, there is no online presentation tomorrow. This is the last one.
So please try. We can wait for next 30 seconds if you have, you can. Then.
He has left the meeting, sir. Actually, I have been told that he was to be giving his yesterday also, and he was coming in and going out. Yesterday also he had some technical issues. So we'll wait for another 15 seconds and we'll then wind it up. So he has left. So
So no problem, Deepthi.
yes, sir.
So we can bind it here.
Yes, sir.
So from my side, thank you, Deepthi, for managing this very well. I also congratulate all the participants that they come up with their work and they have presented very well. And amongst them, I congratulate few of the undergraduate students because I know there's limitations with them, and in colleges, despite of that, they have enthusiasm and they did something towards research, and they presented at last. So my best wishes to all the participants and wish you all the best for the career. And thank you, Deepthi, for managing this very well. Thank you.
Thank you, sir. So I would like to kindly give the formal vote of thanks to everyone. So I stand before you today with the immense gratitude and a heart full of appreciation as we come to the close of this remarkable second day international conference event. It has been an honor to serve as your host and deliver this vote of thanks on behalf of our entire international conference college community. So I would like to speak some lines for you, sir. स्वीकृत आमंत्रण किया, रखा हमारा मान, कैसे करे कृतज्ञता, स्वागत है आपका श्रीमान। अर्पित श्रीमान आपको चंद सुवासित फूल, क्षमा आप करना हमें, हो गई हो जो हमसे भूल। Sir, I would like to
Thank you.
extend my heartfelt thanks to our distinguished chairman, sir, and that you are gracing this occasion with your presence and wisdom. Your guidance and support has been invaluable in making this event a resounding success. Your willingness to lead and inspire our college community is truly commendable, and we are grateful for your time and commitment. I would also like to express our sincere appreciation to all our faculty members, staff, and students who have worked tirelessly behind the scenes to ensure the smooth functioning of the event. Their dedication and hard work have not gone unnoticed, and we owe the success of today's program to their efforts. A very special thanks to all the participants who have contributed their talent and expertise to make this event enlightening. Their contributions have added depth and color to our program, and we are grateful for their participation. I also extend my heartfelt thank you to my student volunteer Porush, who spent countless hours planning and executing every detail flawlessly. His commitment and passion has been instrumental to make this event a memorable event. So once again, I thank you, sir, for your guidance and inspiration, and thank you everyone who has contributed to the success of this event. So we will look forward for more such future endeavors and the continued growth of our college community. Thank you, sir, and thank you everyone, and have a wonderful day. Thank you.
Thank you very much. Thank you,
Thank
Deepthi.
you, ma'am. Thank you, sir.
Thank you. Welcome. You're welcome. So I think there is a
Thank you,
prize
ma'am.
certificate distribution ceremony tomorrow. The schedule has been already been shared with all of you. So who can collect the certificates can collect it personally tomorrow, or the certificates will be mailed on your emails also.
Okay, ma'am.
Okay. Thank you, sir. Thank you again once again.
Okay, thank you. Thank you.
Thank you so much, ma'am. Thank you, sir. Thank you.
Okay, sir. So we may leave now.